Hello, everyone, and welcome to today's webinar, which is going to be devoted to the cardiovascular diseases and the lipidomics of cardiovascular diseases. Cardiovascular disease is an overall term describing diseases of the heart and blood vessels. There are, of course, several risk factors for cardiovascular diseases, and the main of them are, as most of you most likely know, unhealthy diet, lack of physical activity, and tobacco and alcohol use. Today, we're going to focus on several diseases. Of course, we cannot cover all of them. So we're going to focus on coronary heart disease, then cerebrovascular diseases, and congenital heart disease. CVD overall has a huge death toll. According to the World Health Organization, cardiovascular diseases are the leading cause of death worldwide, being responsible for over 30% of death globally. It comes to combined ischemic heart disease and stroke. We do know that lipids play a huge role in the cardiovascular disease development. So I'd like you to very briefly give an introduction to lipids. Lipids are a very heterogeneous group of molecules. They consist of an apolar part, usually made up of fatty acids, and a polar head group. For example, in some lipids, we can achieve structural diversity by different fatty acids with different fatty acid chain length and number of double bonds. Where do we find them? We do see them in forms of, for example, lipoproteins. So lipids are part of our nutrients that come to us with the diet, and once eaten, they enter our bloodstream in structures called lipoprotein particles. And this lipoprotein, these particles, they consist of an outer shell of phospholipids and a core of neutral storage lipids, triglycerides and cholesterol esters. The lipoproteins are the vehicles used to transport lipids to the various organs, where they serve as nutrients, actually, and building blocks for cell structures in the form of membranes. Lipids are relevant for cardiovascular diseases. For example, fatty acids, they play a role in the inflammation, impaired cardiac function. They play a role in the development of insulin resistance, thrombosis, atherosclerosis, and other conditions and diseases. TECs, they play a role when it comes to impaired HDL structure and function and obesity. Also, high LDL play a role in endothelial dysfunction, and low HDL play a role in overall impaired HDL function and also in endothelial dysfunction. Now, when we know that lipids are relevant for cardiovascular diseases, I would like to move towards the particular case studies where we, together with our partners and clients, teamed up to study lipids and cardiovascular diseases. First, we wanted to explore the lipid potential for differential diagnosis of cardiovascular diseases. So we wanted to see if cardiovascular diseases alter plasma lipidome and if we can use lipidomics analysis to differentiate between various cardiovascular disease subgroups. This study resulted in a publication, and we wanted to see, in this case, we used blood lipidome as a generalized measure for the metabolic status. Here is the study design overview. Total of 427 participants were involved in this study, 85 belonging to the control group, and 217 belonging to the group of various cardiovascular diseases, with another 21 participants belonging to the ischemic stroke group and 104 to the systemic lupus erythematosus group. Sample acquisition was followed by shotgun mass spectrometry lipidomics analysis of obtained samples And then we followed up with the lipid identification and quantification, and further data analysis was performed. The analysis of the acquired dataset included several machine learning techniques, and some of them were supervised and some of them were unsupervised. While supervised machine learning relies on labeled input and output training datasets, unsupervised methods rely on unlabeled datasets. For unsupervised analysis, in this case, principal component analysis or PCA was used, and a reasonable separation between, of course, age match controls and ischemic stroke and different groups of cardiovascular diseases was achieved. Well, in some cases, for some cardiovascular disease subgroups, the separation was not too good for some groups. On the other hand, the separation was performed really good. For example, in this case, we see the group 4 and 5 of cardiovascular diseases, and we achieved almost complete separations, suggesting that lipid abundances correlate with the disease presence. Further, we wanted to see what are the significantly regulated lipids within various groups of diseases. So here you see the comparison, this heat map that visualizes the direction of significantly upregulated and downregulated lipids in different disease groups and in cardiovascular diseases. On the right, you see the set of lipids which play the role, which are the main contributors, and having these lipids at hand, we performed further separation of different disease groups based on this particular pre-selected lipids using linear discriminant analysis. So LDA plot, here you can see it, and we see that the separation of CVD groups, subgroups, and control is really good, and we see that this method essentially works to separate cardiovascular diseases subgroups and other disease groups from control very well. Further, we see that both PCA and LDA analysis results, we see that this result suggested building and testing partial least square classifiers, and we created several of them and tested on independent test sets. And we see the classification performance based on a set of lipids showed promising results for cardiovascular diseases versus control. Of course, for some subgroups was better, for some groups were worse, but overall, the CVD separation was over 0.8, varying on the particular subgroup, showing that this method really works well. So, lipidomic profiles of cardiovascular diseases was at the major were major analysis point, and we do know that lipids play an essential role in chronic inflammatory diseases and cardiovascular diseases, and we saw that cardiovascular diseases alter lipidome, and these alterations are reflected in plasma lipidome. Finally, we did confirm that lipidomics analysis allows for the differentiation of cardiovascular disease subgroups. Another study which I wanted to discuss with you is basically called Lipidomics risk scores. And overall, we do know that early and accurate identification of individuals at high risk for both type 2 diabetes and cardiovascular disease is, of course, crucial for effective management and prevention of these diseases. So with this study, we wanted to see if lipidomics analysis can predict the development of cardiovascular diseases, because by detecting these risk factors at an early stage, individuals can adapt their lifestyle, their diet, their physical activity to lower their risk of developing these diseases. In addition, early risk identification can also enable healthcare providers to monitor individuals more closely and offer appropriate interventions and treatments if necessary. And timely detection of type 2 diabetes and cardiovascular disease risk is essential for reducing the disease burden and improving public health outcomes. The results of these studies were also published together with our collaborators and partners and customers. And this is the study design. Essentially, genomic and lipidomic analysis of blood plasma samples of 4,067 participants were performed. We had an information about their clinical tests, lipidome and DNA and SNPs, which I'm going to talk a bit further. And then we calculated their risk scores. We wanted to add SNP information, and SNPs are single nucleotide polymorphisms, which are essentially point mutations in our DNA. And it is known that polygenic risk scores are based on so-called SNPs, and certain SNPs are associated with risk for getting a specific disease. Actually, usually it's a combination of many different of these point mutations that are used to calculate the risk of getting a particular disease. This is so because most diseases are polygenic. And that is why the risk scores based on SNPs actually are called polygenic risk scores. So what we thought is that we could do similar thing with lipids. So we determined which lipids are associated with developing, for example, diabetes or cardiovascular disease in the future. And we use this to calculate a lipidomic risk score in the same way as it's done for polygenic risk scores for each individual in the study. So we then sorted subjects according to the risk score and calculated the diabetes case rate in each decile. In the highest decile, we have a case rate of approximately 25% for the polygenic risk score, and lipidomic risk score results in case rate of close to 40%. Essentially, what it tells us that lipidomics is better than genomics and other potential ways to calculate risk scores. And we wanted to further investigate the effect of lipidome alterations on cardiovascular diseases and type 2 diabetes development risk. So we performed clustering based on the lipidomics data, and then it allowed us to distinguish 6 major subgroups, which had different distributions of risk scores. The clusters were then compared to the risk of type 2 diabetes and cardiovascular diseases, and the group with the highest risk for both was identified. Here it is visualized with the blue color, it's group number 6. And while the highest subgroup risk, highest risk subgroup did not really have any drastic differences from other groups in terms of vital and clinical parameters, this group showed a striking lipidome signature. The concentrations of 9 lipid classes, including cholesterol, certain lysolipids, and others, were significantly altered, while regular and ether-linked phosphatidylcholins, DAGs, and TAGs did not show any significant changes compared to other subgroups. Eventually, so this is a hierarchical clustering based on 148 lipid concentrations, and this is what we saw. To conclude, early disease risk detection allows for timely intervention, and we showed that classical clinical parameters are suboptimal for risk evaluation. What we see is that lipidomic risk scores improve the current ways of risk predicting for cardiovascular diseases and type 2 diabetes. And this study indeed highlights the potential importance of lipidome in predicting disease risk and the possibility of using lipidomic risk scores for early disease risk assessment for both type 2 diabetes and cardiovascular diseases. The results of this population health study suggest that measuring the lipidome regularly over time could help to monitor a person's health status and understand how lifestyle and diet can impact their health. And by identifying these lipidomic alterations early on, it may be possible to intervene and prevent the onset or progression of a disease, or to adjust a lifestyle or dietary habits to reduce the risk of developing a disease in the future. So this could be especially important in the early stages of a person's life when risk assessment is crucial for disease prevention and early intervention. Now, how else can we use additional omics information, which we have already talked about in terms of genomics? There is another study I wanted to show you, and this is the Multiomics also study in cardiovascular diseases. And we wanted to see if multiomic analysis allows to discover new predictive factors for cardiovascular disease development. So the study included participants from the following cohorts: EUFAM, FinRisk, FinGen, and UK Biobank. Plasma lipids such as cholesterol, triglycerides, and lipoproteins are often used to estimate an individual's risk in developing cardiovascular diseases. We just checked it out, and we saw it in the previous case, and we also know this from our regular blood test monitorings. Often, certain cardiovascular and inflammatory diseases are reflected in plasma lipidome. We also have checked it. And while informative, a more comprehensive picture of the plasma lipidome, especially if combined with other omics endpoints, may improve prediction and inform patients and doctors about cardiovascular diseases and other diseases. So this report, which was published, examined the association between genotypes and plasma lipidome and the risk for cardiovascular diseases. This multiomic studies results, they established that the concentrations of certain plasma lipid species are heritable, and that these metrics can be used to predict the risk for cardiovascular diseases and diabetes. Again, the SNPs information was acquired. The cohort included over 2,000 participants, and individual samples were analyzed via genotyping and plasma lipidomics, followed by association analysis. This approach allowed us to identify over 9 million genetic markers, and we measured, identified, and quantified 101 lipid species that were distributed among 13 lipid classes, and we figured out the numerous associations between genotypes and lipid species. For example, we do see that ceramides and lipid-containing PUFAs, polyunsaturated fatty acids, were highly heritable, and phosphatidyl inositols were the least heritable. As in the risk-score study, the genomic differences of the study participants was assessed based on SNPs, and the combination of these omicses allowed us to calculate to what extent lipid levels are genetically determined. For example, one of the major outcomes of this analysis was that the levels of polyunsaturated acids, like arachidonic acid, for example, depend significantly more on genome compared to the rest of the fatty acids. Here you see the fatty acids of 24, 25, and 22,6, and we see that they're much more dependent on the genotype compared to other fatty acids. Another thing that we found out is that particular SNPs have actual phenotypic consequences regarding changes in lipid levels. Here shown an example for some particular SNP and sphingomyelin. This particular SNP is in turn associated with the incidence of cardiovascular diseases. Here you see in red, which is characterized by damaged blood vessels in the brain and resulting in a so-called mini stroke. This shows that the combination of genomics and lipidomics is quite powerful, and it has the potential to identify a causal relationship between genetic markers and a disease state by determining physiological changes in this case in lipid metabolism and associated with a specific genotype. Actually, we could identify many more of these associations, and it has the potential to deliver information to novel drug targets in disease prevention strategies, such as lipid-altering metabolism therapies. Overall, type 2 diabetes and CVD risk subgroups showed to be based on lipidome profile and multiomic analysis of over 2,000 plasma samples found links between genotypes, lipids, and cardiovascular diseases. And this allows us to discover new predictive factors for cardiovascular disease development. And this study studies Multiomics database, which is comprised of populational, genotypic, lipidomic, and phenotypic data, may also serve as a source for the future studies of populational health. Finally, I would like to present the last case study, the last article for today, which is more about translational medicine and translational application. So in this particular study, which I'm going to talk about, they used also a mouse model to see what they're interested in, and then further on applied this knowledge to human samples. This study was taken care and was analyzing heart failure. And what we wanted to know was if cardiac lipid metabolism changes in after heart failure, and are these changes reflected in human lipidome remodeling after heart failure? This study was also published together with many of our colleagues from different hospitals and universities and university clinics and institutes in Germany. Here is the schematic representation of left-sided systolic heart failure. So on the left, you see the normal heart, on the right, systolic heart failure. And overall, heart failure occurs when the organ progressively loses its capacity to pump blood to the rest of the body. And this condition is typically caused by some damage of cardiac tissue through maybe myocardial infarction, coronary artery disease, or some other genetic factors maybe. And patients may experience fatigue, shortness of breath, irregular heartbeat, edema, some metabolic changes, of course, maybe kidney dysfunction. And, you know, it can also include other comorbidities such hypertension, obesity, or diabetes. We do know that cardiovascular diseases, which include heart failure, are the leading cause of death worldwide. Moreover, the global incidence of heart failure is increasing. However, accurate diagnostic, unfortunately, and effective therapies both remain elusive. So, characterization of the lipidome has recently emerged as a tool to identify the risk factors for such conditions. And heart failure is accompanied by an increase in lipid metabolism and in adipose tissue as well, in cardiac tissue and other organs. As a result, the lipidome, which of course is hypothesized to modulate heart function, is altered. Moreover, a growing body of evidence suggests a key role for cross-tissue or organ communication in physiology and pathology. These processes have not been comprehensively characterized in heart failure before this study was conducted. Here is the schematic depicting of a constriction of the transverse aortic arc in mice. This is how heart failure can and is replicated in mice. And following this procedure, mice exhibit signs of heart failure, including altered cardiac tissue morphology, restricted cardiac function, and expressions of genes linked to cardiac failure. Interestingly enough, mice may lack adipose triglyceride lipase, which is called ATGL. And when they lack this enzyme that catalyzes lipid metabolism in adipose tissue, they're somehow protected from such effects of a cardiac failure. And if we compare the the lipidomes of mice with normal and not functioning ATGL, we see that there are quite huge differences. In this case, we were able to measure 225 lipid species from 18 lipid classes. And among them, of course, phosphatidylcholins, phosphatidylethanolamines, and cardiolipids were the most abundant. And in this case, we see that the lipidomics analysis showed that 14 out of 225 lipid species were upregulated in murine cardiac tissue samples. Yet the absence of ATGL weakened these changes in cardiac lipid composition. The largest change was an increase in phosphatidylethanolamine species. And these lipids, they consist of a glycerol backbone attached to one phosphatidylethanolamine and two fatty acids. So high levels of phosphatidylethanolamines have previously been linked to reduced cell membrane integrity and subsequent cell death. And this lipid-mediated process also may be involved in the pathology of cardiac failure. To confirm the discovered lipid composition changes, in mice, lipidomics was applied to human blood plasma samples from male who suffered from left-sided systolic heart failure and control samples, of course. In human samples, we were able to detect 147 lipid species from 13 lipid classes. And in comparison to the murine tissue, phosphatidylcholines, triacylglycerols, and sterile lipids such as cholesterol or cholesterol esters were the most abundant in human blood plasma. In patients with heart failure, 8 out of 147 lipid species were upregulated in blood plasma samples, and 3 of those were phosphatidylethanolamines. And notably, it were the same 3 phosphatidylethanolamine species as in cardiac tissue from the mouse model. This was a very strong indicator for a link between the blood lipidome and cardiac tissue lipidome. Here are these 3 elevated phosphatidylethanol lipid species in both mouse and human following heart failure. And they were elevated in cardiac tissue in mice and in human blood plasma samples after the heart failure. Overall, this study demonstrates that in both humans and mice, the lipidome is altered during and after heart failure. Moreover, the result from murine cardiac tissue suggests that these changes are at least partly regulated by lipid metabolism in adipose tissue, which may essentially represent a therapeutic opportunity. Of course, future work is needed to figure out the underlying mechanism of these observations. To conclude this part, we do know that cardiac lipid metabolism changes in mice after heart failure are there essentially, and experiments with mice show that deleting the potential target enzyme ATGL halt this process. And during this study, we have discovered links between mice and humans in terms of lipidome remodeling after heart failure. With this, I would like to move towards the final slide. I would like to conclude this webinar and actually just once again underline the things that we went through today. First, cardiovascular diseases alter lipidome, and these alterations are reflected in plasma lipidome. It's very important that we see these changes in plasma lipidome, which is the minimally invasive method to see the metabolic state. Lipidomics analysis allows us for differentiation of cardiovascular diseases from other diseases and of cardiovascular diseases, improves the current ways of risk prediction for cardiovascular disease and type 2 diabetes, and multiomic analysis allows to discover new predictive factors for cardiovascular disease development. With this, it would be all from my side, and thank you so much for your attention. See you next webinar.